Hello to our viewers and a warm welcome to the Science AAAS Webinar, Harnessing In Situ Transcriptomics to Reveal Complex Tissue Architecture at Single-Cell Resolution. My name is Sean Sanders and I'm the Senior Editor for Custom Publishing at Science. It's my pleasure to act as moderator for this event. Sequencing of RNA can provide a trove of useful information for quantification and characterization of the transcriptome. The problem with bulk sequencing, though, is that spatial and cellular heterogeneity information is lost. Enter single-cell analysis, which can provide unprecedented detail on cellular diversity in development, health, and disease. This emerging area of spatial transcriptomics aims to map a single-cell level molecular atlas of human tissues in situ. However, mapping entire tissue areas at single-cell quantitative resolution presents significant challenges for imaging and data analysis. In this webinar, efforts to develop large-scale in situ transcriptomics will be described, using the mouse brain as a model system for mapping the molecular diversity of neuronal and glial cell types. The technology that makes this high-resolution mapping possible will be described and compared with other current methods. It is my pleasure to introduce today's speakers to you now. Dr. Omer Bayraktar from Wellcome Sanger Institute in Hingston, UK, and Dr. Xiao-Jun Ma from Advanced Cell Diagnostics in San Francisco, California. Due to scheduling challenges, we have pre-recorded the presentations from both of our speakers. Dr. Bayraktar is in the midst of moving his laboratory from the US to the UK, but he will be on the line later to answer questions following the presentations. Dr. Ma is traveling in China, but in his place we have Dr. BQ Zhang, also from Advanced Cell Diagnostics, who will be able to answer any questions about his presentation. Before we get started, I'd like to share some information for our online viewers. At the top right of your screen, you'll find photographs of today's speakers and a View Bio link, which you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. After the speaker presentations, we will have a short Q&A session, during which we will address some of the questions submitted by our live online audience. So if you're joining us live, start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Advanced Cell Diagnostics for sponsoring today's webinar. Now I'd like to introduce our first speaker, Dr. Omer Bayraktar. Dr. Bayraktar is a new group leader at the Wellcome Sanger Institute in Hingston, United Kingdom. He's interested in large-scale mapping of cellular diversity across human tissues with a focus on nervous system development and disease. He was a postdoctoral fellow at the University of California, San Francisco, and the University of Cambridge, where he developed a high-content in situ transcriptomic pipeline for mapping large tissue areas. He used this approach to elucidate the heterogeneity of astroglial cells across multiple layers and different areas of the mammalian cerebral cortex. I'll now play the recording of Dr. Bayraktar's presentation.
Thank you very much for the invitation, Sean. So as Sean introduced me, my name is Omer Bayraktar, and I'm a new group leader at the Wellcome Sanger Institute at Hingston in the UK. I'm interested in exploring the cell type diversity in the brain, and today I'll tell you about my work on this area using new in situ spatial transcriptomic methods that allow mapping gene expression across tissues in situ. I'll start by giving you an introduction to these spatial gene expression methods and why we need them. Then I'll tell you about my work in Cambridge as a postdoc on developing a new imaging-based pipeline for large-scale single-cell mapping of tissues and how I use this method to discover the heterogeneity of a poorly understood brain cell type, the astrocyte. And at the end, I'll finish with a few remarks on my future interests and the state of the field. My research is focused on exploring cell type diversity in the brain and understanding how it comes about during development. I'm interested in this area because our brains contain a remarkable diversity of cell types. There are many different types of neurons with distinct morphology, as you can see on the left side, and gene expression as well as electrophysiology. These diverse neuronal subtypes are supported by several types of glial cells, such as astrocytes and microglia, that perform important functions throughout development as well as adulthood. Our minds are built on this rich cellular diversity of the nervous system, and as you would expect, many neurological disorders affect specific brain cell types. To understand what these cells do in the healthy and the diseased brain, we need to identify and map these cells. One way of doing this is using transcriptomic methods to determine the gene expression signatures of neural cell types. As most of you are well aware, the past 20 years have seen an explosion of transcriptomics. Most of these methods dissociate tissues to purify cells of interest. We now have an incredible amount of data describing cell type specific gene expression patterns in healthy tissues, as well as gene expression changes in diseases. Single cell RNA-seq is becoming more robust, pretty much like every week. We can sequence larger and larger cohorts of cells at lower costs. In contrast, the alternative approaches that we use for mapping these transcriptomic findings back into the tissues, mainly in situ hybridization and immunohistochemistry, have not improved at the same rate. The new area of spatial transcriptomics aims to fill this gap, and to this emerging field, there are two immediate applications. First, we can use these alternative in situ methods to validate the transcriptomic findings. Second, beyond just molecular validation, spatial transcriptomic methods allow us to study the cells in their native environment and the intact tissue. This way, we can resolve spatial relationships between cells, for example, stem cells and supporting meat cells, and also determine the functional global organization of tissues. In my research in spatial transcriptomics, from a technical standpoint, my aim has been delivering a robust pipeline for large-scale in situ mapping. I'm interested in this for three kinds of long-term applications. First, creating a single-cell level atlas of mammalian organs. We can combine single-cell RNA-seq and in situ methods for full spatial transcriptomic reconstruction of diverse tissues across the human body as well as model organisms. Second, once we map the healthy human cell atlas, we can move on to the sick human cell atlas. We can use these spatial methods to decipher cell type specific gene expression changes in various diseases. Last, I think clinical use is on the horizon for in situ spatial mapping. Pathology labs are definitely no strangers to in situ hybridization, yet imagine robustly assaying 10 multiplex biomarkers across pathological specimens at high resolution. With automated imaging and analysis, we can have the next generation of molecular diagnostics. And while you can apply spatial transcriptomics to many tissues, one of the biggest challenges across the mammalian bodies is the incredible cell type diversity in the brain. To map the cellular diversity in the brain, spatial transcriptomic methods have to satisfy three massive technical requirements. First, in the brain, many distinct cell types are located right next to each other. So to be able to distinguish them, we have to do high-resolution imaging. This high-resolution imaging needs to be performed for very large tissue areas. As the brain is very large in size and regionally diverse, you need to be able to image large areas of tissue, brain tissue, through clearing or fast imaging of histology sections. Second, we know that all of these different brain cell types, neurons and different types of glia, are distinguished by the expression of a large number of genes. So any pipeline has to be able to screen large number of genes across large tissue sections. Last, high content data such as many genes and high throughput data such as large tissue area high resolution scans generates very, very big data. So our technical pipelines need to be able to process this large image data in a user-friendly format for the most application. So what are the available tools for spatial transcriptomics? The hybridization-based approaches are the most developed, and I'd like to give you a very quick lay of the land. The most widely used approach is single molecule fluorescent in situ hybridization, or SMFISH for short. Here, we're using multiple fluorescently tagged probes to hybridize them to an RNA target in vitro or in vivo. The RNA molecules are visualized as distinct puncta. These spots are thought to be single molecules. This approach allows us to quantify single-cell gene expression from images using segmentation and spot detection methods, but it suffers from high background fluorescence on tissues. The upgrade on this is amplified SMFISH, which basically tags these RNA labeling probes with a lot more fluorescent or enzymatic labels to improve the signal. There are a couple variants of this signal amplification. Today I'll be talking to you about RNA-scope SMFISH, developed by Advanced Cell Diagnostics, which uses a branched single-stranded DNA probe complex for signal amplification. I'll leave the technical details on this to Xiao-Jun Ma from ACD, whom you will hear from next. Yet, suffice to say that the RNA-scope works robustly on tissue sections, but it has not been used large-scale in vivo. So these now standard SMFISH methods allow you to look at 4 to 5 genes in a multiplex manner, but recent years have also seen the proliferation of highly multiplexed in situ hybridization and sequence technologies. So these new methods allow you to use multiple cycles of probe hybridization, imaging, and probe stripping to visualize many, many genes at the same time. For example, in one of these newer approaches called sequential barcoding fish, or SecFISH for short, you use a temporal barcoding strategy where the aligned combination of RNA spots from every cycle is unique to each gene. This allows you to assay more than 200 genes, but it requires complex experimentation and analysis to align millions of spots across images. Another barrier to scaling this approach to large tissue areas or, say, human tissue sections is imaging, which takes a long time at high resolution for covering large tissue volumes. In addition to the hybridization methods, there is a number of array-based spatial transcriptomics, which are highly promising in terms of their throughput, but currently lack single-cell spatial resolution. Last, in addition to these hybridization and array-based methods, there is also now a number of in situ sequencing-based technologies, such as Ph.D. or StarMap. While these approaches are also very promising in the future, they're not quite ready for large-scale prime-time application on tissues. So as you can see, there is a number of technologies, but which one is the best for the brain? Right off the bat, looking at what's available, we realized that most of the approaches had automation and imaging bottlenecks, limiting their application to large tissue areas like the brain. So as an alternative, we wanted our pipeline to deliver large-scale, high-throughput output and satisfy the brain cell type gene expression requirements that I mentioned before. So this led us to adapt and combine a number of existing technologies to deliver our new pipeline. We called this LASMAP for large tissue area spatial transcriptomic mapping. All of this work was done during my postdoc in David Rowich's lab at the Wellcome Trust MRC Stem Cell Institute at the University of Cambridge, as well as the University of California, San Francisco. Again, we've named this LASMAP for large area spatial transcriptomic mapping. In this pipeline, we use standard thin tissue sections on normal glass slides. We automate all steps in 4-color RNAscope SM FISH and combined antibody staining. We then use an automated spinning disk confocal for high-resolution imaging of entire tissue sections. We use an image analysis program that can handle large data to identify single cells, to segment single cells in our image data, and count RNA puncta within each cell. Last, we can go into this image data, manually identify different brain regions, and analyze single-cell regional expression patterns across the brain tissue. So now I'd like to quickly run you through some of the technical specs of this pipeline. I'll briefly talk about the components. To automate the histology on SM FISH and immunostaining, we use the LicaBond RX robotic stainer. This is a clinically approved system used in many pathology departments. Here we use standard tissue sections on slides, usually about 16-micron thick cryo sections. Using the stainer, we automate all steps in 4-color RNAscope and subsequent immunostaining. Here we can process about 20 slides in a single run with 10 probe mixtures. So that means that we can screen about 40 genes with 2 replicates. A single run of this, a single staining cycle takes about 14 to 17 hours. So this is something that could be done in the lab overnight. In terms of SM FISH, we use a version of RNAscope that utilizes both branched DNA and termite signal amplification to boost the RNA signal. As a result, we get really high signal-to-noise ratio that allows us to do very fast imaging. In terms of the details of the protocol, I'll let Xiao-Jun Ma to explain the rest. After we automate pathohistology, single molecule FISH and antibody staining, we use an automated spinning disk confocal that can take images really fast, and it can deal with large image data. The system that we use for this is the Operetta CLS from PerkinElmer. This Operetta system was originally built for automated high-content in vitro screening, and we ended up adapting some of these plate-based screening properties to the tissue application. In our pipeline, we go about automatically identifying tissue sections on the glass slides. We start by taking a low magnification scan of the entire slide using DAPI. From these low magnification DAPI images, using image thresholding, we could locate the tissue sections across the X and the Y planes. And then after this tissue identification, we can switch to an automated 4DX water objective that can take multi-6-color Z-stacks at submicron resolution. After the imaging, we go about identifying segmenting single cells across entire tissue sections and quantifying the number of RNA functa. Importantly, the software that runs the imaging and the acquisition harmony is both present on the microscope. So our large image data, which grows into terabytes across days of image acquisition, does not have to leave our original system that we're using to take the images. So to validate our approach, we examined the rich neuronal diversity in the mouse cerebral cortex. On the left, you can see a 3D diagram and a coronal section of the mouse brain where the cortex is outlined in the blue in the middle. A fundamental structural feature of the cerebral cortex is its organization into areas and layers. The cortical areas mapped on the other hemisphere of the section contain neuronal circuits that are functionally specialized for different sensory and cognitive functions, such as vision and hearing. Each cortical area shares a layer organization shown on the right. Superficial to deep, there are 6 neuronal layers that contain distinct neuronal subtypes. These layer neurons are well recognized to be molecularly and anatomically distinct, and they play different parts in different cortical neuronal circuits, as summarized on the right. So what you're looking at here is the validation of our last mile pipeline by the mapping of the expression pattern of 4 known markers of neuronal layers in the mouse cortex. On the top, you can see a low magnification image of a 2-week-old mouse brain hemisection with RNAscope against markers, 4 markers of upper, mid, and deep cortical layers. This entire section was imaged at the resolution shown in the bottom panel with an automated 4DX objective as a 1-micron step-size Z-stack. As you can see, as on the right, as we travel through this Z-stack, we can identify cells, single cells indicated by the yellow arrow, appear and disappear. In our analysis, we obtain multiple Z-stacks across each position. Then we perform a maximum Z projection and work with that 2D data. At the high XYZ resolution we're using for these scans, we end up taking 40 to 50,000 images per tissue section. If we were using standard confocal microscopy, this would take a very long time, yet owing to the fast imaging with the use of a spinning disk, we can complete the scan under 2.5 hours, meaning that we could scan many brain slices in a single day. So what we go about doing here is that after we collect these large image data sets, we identify single cells. In this case, we have used the antibody staining against a neuronal marker, NUN in dark blue, as well as staining for DAPI, which is a nuclear marker, to segment single neurons. Our pipeline basically looks something like this. We can identify the neuronal cell bodies, which contain a lot of RNA, using supervised texture segmentation and image thresholding. After we do this, our pipeline identifies a mixture of single neurons as well as Z-overlapping cell doublets, triplets, and cells that are only partially included in the tissue sections. To filter the doublets and the partial cells, we can use a linear classifier-based machine learning approach where we basically select the single cells that we liked. We provide a training set of 100 cells, and then we let this machine learning algorithm process all of our data and filter the single cells from this mixture of doublets and the partials. Once we have identified the single cells, we go about counting the number of RNA spots for each of the 4 gene species in our cells of interest. So what you're looking at on the right is now a slice of the somatosensory cortex, the area that is marked by a dashed line box on the left. On the slice of the cortex, qualitatively, you can see that the expression patterns of these 4 genes indicates 4 different layers. The diagrams on the right side now show single neuron level quantification of this data. Each spot in these diagrams corresponds to an automatically identified neuron. In each of these diagrams, they are colored quantitatively according to the number of RNA molecules that they express for each gene. And as you can see, qualitatively, these automatically identified expression patterns match what we know about the expression of these genes. So what this tells us is that we can use this automated pipeline to image large areas of tissue and then quantify single cell gene expression. And we were interested to see whether we can use this approach to then identify neuronal subtypes based on the expression of these 4 different markers in an unbiased manner. So to be able to do this, we mapped the expression of these 4 genes using last map over 10 tissue sections from different parts of the brain. This data set, consisting of 10 brain sections, totaled to 300,000 pictures. The size is around 500 gigs, and with our fast imaging, we can actually complete the image acquisition under a day. From these 300,000 pictures, we can go about identifying 80,000 neurons across different parts of the cortex. After cell identification, we go about manually assigning these neurons to different cortical areas, such as the motor or the somatomedial cortex or the auditory cortex, as well as the layers, superficial to deep. We do this based on anatomical landmarks and reference atlases. And after we assign these cells to different brain regions, we can then use dimensionality reduction and clustering approaches to be able to identify molecular subtypes based on the expression of these 4 genes. What you're looking at here on the left is now the subtype analysis of this data. On the very top, you can see that our analysis have identified 10 distinct molecular combinations of these neurons. The violin plots in the middle show the expression patterns. What you're seeing on the right is now just an image of a few of these sections and the diagrams of the cortical areas. As you can appreciate, since we have identified all of these neurons from image data, we know their exact spatial position across the brain. So we can, in a quantitative manner, go about mapping their distribution across cortical areas and the cortical depth, which are shown in the bottom left. So when we do something like this, we can then go about mapping these distinct molecular subtypes back into the tissue and generating a spatial transcriptomic reconstruction as shown in the top right. When we do this, we find that our approach indeed validates many known neuronal subtypes that are present across layers and areas. And in addition, we have discovered new area and layer restricted subtypes. The subtype that's marked by high expression or ROR-B and lower expression of other genes, indicated in the spatial distribution by a red box on the left. We find that it shows an area enrichment to the somatosensory and the visual cortex. And in addition to its area enrichment, it's also restricted in its distribution to layer 5 in these select cortical areas. So taken together, what I've shown you today summarizes the progress we have made in mapping, in developing a pipeline for automated large tissue area mapping, and how we can use this to validate known gene expression, regional gene expression patterns, as well as make new spatial findings. So at this point, I'll switch gears and move from the technical aspects of this pipeline and the validation study to discovering the novel heterogeneity of a poorly understood brain cell type in the brain, the astrocytes. So far I've told you about the neurons. They're an incredibly rich, incredibly interesting cell population, but on their own, neurons are utterly useless. For their many diverse functions throughout development and adulthood, they need support, they need the help of a vast array of glial cell types. Perhaps the most abundant glial cell type is the astrocytes. It constitutes 20 to 40% of all non-neuronal glial cells. Traditionally, astrocytes were only considered as passive brain glue that provided homeostatic support for ion balance, as well as providing nutrients to neurons. However, over the last 15 years, there has been a renaissance in our understanding of astrocytes. Using the newer generation molecular and morphological tools, now we understand that astrocytes are master regulators of synaptic development. Astrocytes are very important players both in synapse, in the promotion of synapse formation, as well as their subsequent refinement. We now know that every synapse in the living brain is actually covered by astrocyte processes that talk back to the neurons, creating a newer concept of tripartite synapses. And finally, all of these roles in brain development and physiology are, as we appreciate now, coupled to roles of astrocytes in neurological diseases. In every single neurological disease that's been assayed, it seems that there is a role for astrocytes. So while we now appreciate that astrocytes are very important cells in the brain, we lack a fundamental understanding of astrocyte heterogeneity. While astrocytes are important, the question is, are astrocytes from different parts of the brain specialized to different functions? So in my research, I chose to pursue this question in the cortex. As I showed you in the first part of this presentation, it's well recognized that neurons are molecularly and functionally distinct across cortical layers. My question was simply, do we see a similar layering of astrocytes throughout the cortical gray matter? So to go about answering this question, I manually purified upper and deep cortical layer astrocytes from the mouse cortex and then performed RNA-seq profiling to identify potential molecular differences. And indeed, when I did this, I surprisingly identified a number of genes that distinguish upper and deep layer astrocytes. As the study is currently in progress, the genes are identified, but I hope you can appreciate that a number of genes here distinguish upper and deep layer astrocytes. X and Y show enrichment in their expression in the upper layers, while Z and Q are enriched in deep layer astrocytes. So this shows you that astrocyte gene expression is distinct across cortical layers, but also raises many questions. For example, are there only two superficial and deep astrocyte layers? What is the actual precise distribution of astrocyte layering through the cortex? In addition to layer differences, are astrocytes distinct across cortical areas? And if astrocytes are indeed distinct across layers, how does this astrocyte heterogeneity come by during brain development? Are astrocytes intrinsically programmed to be different, or is their heterogeneity specified by external cues, such as from layer neurons? So all of these are very interesting questions to me, and for the interest of time, I'll only focus on the first question, which is mapping the precise distribution of astrocyte layers. So to be able to look at astrocyte gene expression with SM-FISH, I used an RNA probe against a highly expressed astrocyte marker, glassed in green. On the left, you can see a low magnification image of a brain section showing that glass labeling is present abundantly throughout most brain regions. On the right, you can see a high magnification image of a cortical upper layer where, using glass and DAPI, we can go about segmenting the astrocyte cell bodies, including their soma, as well as their main processes. When we compare this labeling to what we see for neurons, identified by neuron immunostaining here in blue, or the SIT1 SM-FISH, which is a highly expressed neuronal marker, when we do this, we see that glassed single molecule FISH gives specific labeling of astrocytes. For the interest of time, I'll skip the additional steps that we take to filter for single astrocytes, overruling doublets and triplets, and also methods that we use to ensure that glassed SM-FISH specifically gives us the astrocyte gene expression, namely subtracting neurons and other identified non-astrocyte cell nuclei from the glass positive cell area. So what we've done is that we took, earlier on I told you about this RNA sequencing that we did that suggested that astrocyte layers, upper and superficial, were molecularly distinct. We took 50 of the top differentially expressed markers of layer astrocytes, and then we mapped their expression across the mouse somatosensory cortex with our last map pipeline over several tissue sections and several biological replicates. On the right, you can see the in situ expression plot across cortical layers, where we have binned astrocytes across cortical depth from the superficial to the deep layers. And indeed, you can see that this approach has identified a number of upper and deep layer astrocyte expressed genes. Many of the genes that we identified here showed slight enrichment patterns to the slight biases to the upper layer astrocytes, yet we can detect their expression in most of the astrocytes throughout cortical depth, suggesting that they are more likely to be pan-astrocyte genes as identified, as indicated in the middle. Yet, even with this, we could identify a small number of genes that seem to be specific to upper and deep layer astrocytes. We then went about using our cell type specific expression measurements to further filter these upper and deep layer astrocyte candidates for astrocyte specific expression. When we did this, we indeed observed layer astrocyte enriched expression patterns. What you're looking at here is a section of the somatosensory cortex where you can see on the left that blast marks astrocytes across cortical depth, yet our upper layer astrocyte marker in magenta, its expression is, its localization is enriched to astrocytes only in the upper layers from 2 to 4. You can also see this when you look into the high magnification images of our of single astrocytes. This marker in this upper layer astrocyte marker in magenta is highly abundant in upper layers 2 to 4, but its expression is notably absent from layer 5. So to be able to map the precise spatial distribution of astrocyte layers and compare them to neurons, we then went about quantifying this layer astrocyte gene expression patterns at the single cell level. Now what you see on the right is a single astrocyte level map of a slice of the somatosensory cortex where astrocytes are colored according to the expression of our upper layer astrocyte marker. And indeed, you can see this pattern, this broad gene expression pattern that spans multiple neuronal layers with enrichment in the superficial part of the cortex. We can illustrate this data in a couple of different ways, such as this single astrocyte gene expression plot that is colored according to different neuronal layer positions. When we go about doing this for all of our layer astrocyte markers, we identified that broad gene expression gradients distinguish astrocyte layers to 4 spatial bins. We can observe superficial, mid, and deep layer astrocyte zones in the cortical gray matter from layers 2 to 5. And in addition, we identified that the astrocytes in the deepest cortical layer, layer 6, share gene expression such as the expression of ID3 with the underlying white matter, confirming previous observations that these deep layer astrocytes are similar to those fibrous astrocytes found in the white matter. You can sort of appreciate that these broad astrocyte, these superficial, mid, and deep astrocyte layers that we identify do not necessarily correspond to what we have identified for neurons, shown here as single cell plots and also as interpolated smooth expression plots here. I hope you can appreciate that while neuronal gene expression patterns here are mapped precisely to this sharp neuronal layer boundaries, the astrocyte layers exhibit as broad gradients, and that have diverged from what we are seeing on this classical neuronal architecture. So that's illustrated in here. When we look at neurons, we see these well-defined multiple neuronal layers with sharp architectural limits, cytoarchitectural and gene expression limits. In contrast, we think that astrocytes in both the developing and the adult brain exhibit as different divergent layers that span multiple neuronal laminae. So to sum up what I've told you today, I presented you data on the development and the validation of a new spatial transcriptomic pipeline, LabMap, for mapping large tissue areas. Using this pipeline, I showed you that astrocyte gene expression is diversified throughout cortical layers, and these astrocyte layers are divergent from neuronal laminae. An important biological, perhaps the most important biological conclusion is that cortical structure is not solely composed of neuronal layers. The combination of neuronal and divergent astrocyte layers define a more complex cortical architecture than was previously thought. So I'll close by a few remarks on my future research interests. A large goal for my lab and the community in general is applying large-scale spatial transcriptomic approaches to mapping human organs. This is some work that I am planning to focus on the brain as well as other human organs throughout my ties to the Human Cell Atlas at the Sanger Cell Institute. And in terms of some of the technical benchmarks, I can tell you that the direction that we're really interested in is improving the multiplexing of our approach from 4 genes to many genes. As we add more content, as we make our mapping higher content by adding multiple cycles of imaging, we would like to compensate by increasing our imaging speed, and for this, I'll be using newer generation spineless confocals. While we're still interested in imaging standard tissue sections, the technologies are now allowing looking at applying these mapping approaches to the intact tissue, so you can actually identify 3-dimensional interactions between different cells in thicker tissue volumes. Finally, I think all of the sort of technical benchmarks here will bring added analysis requirements. So at Sanger, I'll definitely focus on automated large-scale as well as 3D analysis approaches. So with that, I'd like to conclude by acknowledging my postdoc mentor, David Rovich, and the lab where all of this work was performed. We also had a great team of collaborators from UCSF, UCLA, and Cambridge. In addition, I'd like to thank funding sources, especially the Life Sciences Research Foundation that sponsored my postdoc research. And last, I'll close by saying that I'm starting my own research group at the Wellcome Sanger Institute, focusing on human spatial transcriptomics and human brain cell type diversity. We have several positions available, and please get in touch with me if you're interested. With that, I'll pass the baton to Xiao-Jun Ma, and then we'll take your questions.
Thank you for that, Dr. Bayraktar. Our next speaker for today is Dr. Xiao-Jun Ma. Dr. Ma is the Chief Scientific Officer at Advanced Cell Diagnostics, where he led R&D for RNAscope and BaseScope technologies for highly sensitive and specific RNA in situ hybridization used in RNA analysis with cellular resolution. He was previously Vice President of R&D and Biostatistics at Bio-Techne and also held positions at Johnson & Johnson and Monsanto Searle, which is now Pfizer. Here is Dr. Ma's presentation.
Thank you, Xiang, for a great introduction, and also thank Omer for a beautiful example showing how RNA-scope can be used for in situ transcriptomics. So RNA-scope is a in situ hybridization technique. RNA-seq has been around for more than 40 years. RNA-scope is different from other methods in its probe design strategy, as shown here by a pair of Z's. We call these double Z probes. Double Z probes couple target-specific binding and signal amplification, such that only when a pair of Z probes bind simultaneously to a target, signal amplification can occur. This greatly reduces the background and improves signal-noise ratio by 400-fold. Single RNA molecules are detected as punctate dots, as you can see here. You can quantify your target RNA by simply counting the number of dots per cell. In addition, a pair of double Z probes bind to a target sequence of in about 40 to 50 nucleotides in length. This makes the assay compatible with partially compromised RNA as those found in clinical FFPE specimens. Here is a overview of the RNA-scope assay. And you start with target retrieval, which unmasks the target RNA in the tissue and cells. And then you hybridize the double Z probes to the target RNA molecule. And then you amplify the hybridization signal through sequential hybridization with amplifiers and label probes, which builds a branching-like tree at your target molecule, which can then be visualized under a standard bright field microscope or fluorescent microscope. You can quantify your target expression by counting the dots manually or using automated imaging analysis software. The RNA-scope assay was launched commercially in 2011. We have been continuously improving and expanding its capabilities. And nowadays, you can run the assay in many different configurations, chromogenic or fluorescent, automated or manual, single-plex or multiplex. The most recent addition to our product line is the BaseScope assay. BaseScope assay further improves the RNA sensitivity and specificity to detect sequences as short as 50 bases and discriminate single base differences. BaseScope assays make it possible to analyze alternative splicing variants, circular RNAs, detect a specific member from a highly homologous sequence family down to point mutations. In the multiplex fluorescent assay used by Omer, we added additional layer of signal amplification by using TSA conjugated fluorescent dyes. This generates very bright signals even in FFPE tissue. Let's take a look at this protocol in more detail. The entire RNA-scope multiplex assay protocol is fully automated on the Leica-bound system and has the following steps. You start pre-treatment from baking slides to DOX to target retrieval, protease digestion to open up the target for hybridization. And then you hybridize your target probes simultaneously and also amplify your signals simultaneously. And then you detect your targets sequentially one at a time using different TSA fluorophores, which is followed by staining with DAPI for of the nuclei. Because HRP is used to amplify the fluorescent signals, it is essential to block the HRP activity between each detection step. So HRP from one run does not contribute to the following run. This is some data that demonstrates that we have a very efficient and complete HRP blocking step. This was demonstrated using a 3-plex assay, detecting highly abundant positive gene UBC in channel 1, and our negative control probe, that be in channels 2 and 3. As you can see here, there is no spillover of signals from the highly expressed UBC1 to the two other channels. Another question is that whether high expressors might mask the signal from low expressors. So we challenged our system by doing a 3-plex assay for the highly expressed highly abundant 18S ribosome RNA, which is present in millions of copies per cell, and then the highly expressed UBC in channel 2 and relatively lowly expressed gene polar 2A. As you can see here, the polar 2A signal from this three-plex assay was comparable to that from a single-plex assay, confirming that there's no masking of low expressor signals by the high expressors. The sequential target detection protocol makes it possible to combine RNA-ish with protein detection by immunofluorescence. For example, in channel 4, instead of detecting an RNA, we incubate the slide with a primary antibody for your target of interest and then proceed as you would for regular IF. So how does the result look? This is an example taken from a publication by one of our customers published in Cell last year. They were trying to categorize the 3 distinct subpopulations of prelimbic neuron projections. They used the GFP antibody staining to identify the 3 neuronal populations and used RNA-scope to detect 2 mRNAs, TNNC1 and POU3F1. By visualizing GFP and RNAs together, we can see that these anatomically distinct neuronal populations actually are different at molecular level, as the expression pattern of 2 mRNAs, TNNC1 and POU3F1 are different in the 3 regions. Here's another example of detecting 3 RNAs and 1 protein in the tumor microenvironment of non-small cell lung cancer. The 3 RNAs are immune checkpoint markers PD-1, TIM3, and PD-L1. We detected the CD45 protein by using antibody, shown in white. As you can see here, with the CD45 positive staining, we can see PD-1 and TIM3 expression in a subset of CD45 positive immune cells. We have been thrilled by the speed and scope of adoption of the RNA-scope technology by researchers all over the world. This is best illustrated by the publications by our customers. RNA-scope has been featured in every top journals in the scientific community. Now it has the RNA-scope publications is coming out at almost 2 papers per day. As shown in this graph, the RNA-scope has been used very broadly in many fields of research, with cancer, neuroscience, infectious diseases being the top 3 applications. To summarize, RNA-scope uses a unique probe design strategy to dramatically improve signal-to-noise ratio. We currently have over 15,000 probes on our catalog. New probes can be designed and made within 2 weeks for any target and any species. RNA-scope provides both quantitative and spatial information of your target expression in complex tissue environment at single-cell resolution. The newest assay, BaseScope, can detect splice variants, short sequences down to 50 bases, can discriminate highly homologous sequences down to a single base. RNA-scope assays can be complemented by immunohistochemistry or immunofluorescence for simultaneous RNA and protein detection on the same slide. I will stop here. Thank you for listening.
Thank you so much, Dr. Ma. We're now going to move on to the questions submitted by our live online viewers. A quick reminder to those watching us live that you can still submit your questions by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. Before we get to the questions, I wanted to welcome Dr. BQ Zhang, who will be sitting in for Dr. Ma to answer questions. And of course, we also have Dr. Bayraktar on the line with us. So the first question I'm going to come to is for you, Dr. Bayraktar. How does LastMap compare with StarMap in terms of sensitivity, time, and the number of genes that you're able to probe?
Yes, that's a great question. So currently a direct comparison is lacking since both LastMap and StarMap are brand new methods. We have not been able to do the side-by-side comparison on the same tissue in the same lab. But in terms of their multiplexing, currently our approach is restricted to 4 genes, while StarMap is focused on higher multiplexing from 16 to 160 to 1,000 genes. The main difference between our method and StarMap is that our method is focused on mapping large tissue areas. So we can go about applying this to entire mouse brain sections to look at gene expression differences across very large areas, large population of cells, or we can go about assaying entire human tissue sections at single cell resolution. We can do this because we're using fast imaging and integrated fast data analysis. StarMap, while it provides high content data in terms of the number of genes you're assaying, its imaging is limited to small tissue areas. So in the future, I think one of our goals will be improving the multiplexing of LastMap to be able to assay more than 4 genes. While I can see similar developments happening in the StarMap in terms of improving their imaging speed.
Thank you for that. So let me come to you, Dr. Zhang, with this question as well, together with another related question that says that RNAscope is apparently limited to 4 genes per assay, and there's not a stripping and restaining procedure. Are you aware of any advances that might allow more than 4 genes to be probed in a single assay?
Yeah, that's a great question. We actually hear a lot this question many times from our customers and collaborators. So we are currently working on a new product which will allow detection of up to 12 targets simultaneously, and this is our first phase of this product. This one will utilize some cleaving and rehiving procedures because it's just the pure limitation of the fluorophore spectrums. But with that, we will be able to achieve 12 genes on the same section.
Thank you very much, Dr. Zhang. So let me stay with you with a question that asks, what is the sensitivity of detection for this assay, and can cells with rare RNAs between 10 and 100 copies perhaps be detected?
Yeah, so RNAscope offers very high sensitivity amplification. We have, so especially with what Omer just talked about, the LS multiplex or our manual V2 assay, we have the addition of the TSA amplification system, which will allow the visualization of very few copy numbers in the cell. So 10 to 100 wouldn't be an issue for us. And each dot you see will be a single copy of RNA present in the cell. And by quantifying those single dots, you can get an estimation of how many copies of the transcript is present in the cell.
Excellent. Thank you very much. So we're almost at the end of today's webinar, but I'd like to get in a couple more questions. Dr. Bayraktar, in view of the extensive dendritic network of single neurons, this viewer asks, to what extent does examining RNA in the cell body represent RNA expression in the entire cell, including the dendrites?
Yes. So in our study, we actually intentionally focused on quantifying RNA in the neuronal cell bodies because to be able to attribute, RNA molecules to the cell body or the dendrites, you need to be able to segment those cellular compartments accurately. Right now on mouse tissue sections, we were more confident of our ability to segment neuronal soma, the cell bodies. But I think that if you have good markers of dendrites, and if you can precisely quantify segment dendritic processes through thin or thick tissue sections, there should be no impediments to quantifying their dendrite localization.
Let me give you one final question before we close, and we have received a couple of queries on this topic. Is there any reason that the technique that you're using could not be applied to other mammalian brains, particularly human, and what might the challenges be to doing this?
Mm-hmm. So we have actually designed LastMap with large tissue area screening in mind. So I think amongst all of the available single molecule fish or in situ sequencing options, ours is the best option for looking at large tissue areas.
yet there will be a couple of problems when you're dealing with human tissue sections. First, the amount of data, the data acquisition length, and the data size that will be generated will be much higher. So, for efficient screening, you need to be able to scale up your imaging operation by using additional systems. And second, with human, especially adult human brain tissue, there are technical problems associated with fluorescence detection, namely the autofluorescence of lipofuscin molecules that are enriched in aging brains. So in our hands, we've had some luck with using the TSA amplification to overcome the lipofuscin autofluorescence, but obviously there is lots that can be done in terms of streamlining and optimizing human tissue preparation, preservation, and fluorescence detection methods.
Thank you so much, Dr. Bayraktar, and we're unfortunately going to have to end the webinar there. But before we go, I wanted to thank today's speakers once again, Dr. Omer Bayraktar from the Wellcome Sanger Institute, Dr. Xiao-Jun Ma from Advanced Cell Diagnostics, and Dr. BQ Zhang, also from Advanced Cell Diagnostics, for helping answer the viewer questions. Please go to the URL that will now be appearing at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation within about 48 hours from now. We're interested to know what you thought of the webinar. Send us an email at the address now up in your slide viewer, webinar@AAAS.org. Again, thank you so much to our panel and to Advanced Cell Diagnostics for their kind sponsorship of today's educational seminar. Goodbye.